Hello and welcome to our lecture about protein structure. Today we are going to be learning about protein structure and the effect of protein shape on protein function. Alright, so just a review. Remember that proteins are polymers of amino acids. The amino acid, of course, is the monomer. There are 20 different amino acids. I'll show you a slide of that in a moment, but all have the same four basic parts, right? There's the amino group here, the carboxyl group at the other end, the R group up here, and the central carbon. And remember also that we can sometimes see this ionized where we don't have this hydrogen here and instead we have a negative charge. And over on the amino group we have another hydrogen with a positive charge. And then to build a protein we link a bunch of these together using our friend dehydration synthesis. All right, here's showing that slide with all the different amino acids with their different R groups. Notice they're shown in their ionized form. And of course, what we really want to notice with this is that every amino acid has the same basic structure of the amino group, central carbon, carboxyl group, and then that R group attached. The R group is in the white box. The R group is unique and lends different character to each one of the amino acids. Every amino acid has its own name and its own three-letter code. This is important not because you are expected to remember them or memorize this, but they need to recognize that every amino acid is different and that the R groups have different characteristics. So these ones here are put together because they are hydrophobic. Okay, we know they're hydrophobic because when we look at their structure, we see either only carbon-carbon and carbon-hydrogen bonds or mostly carbon-carbon and hydrogen bonds. There's only two that have anything different. There's nitrogen there and a sulfur here. But still, they have enough non-polar bonds to make them act like a non-polar molecule. Okay, these ones down here are the opposite. They are hydrophilic. Okay. And if you look at them, they have a lot of polar bonds, right? They have oxygens all over them. Okay, and those oxygens, or sulfur I should say too, also makes it very polar bonds, right? So they can interact with water nicely, and they do. And then these ones over here have a positive charge on them in the R, oops, that one doesn't, right there, in the R group, and these here have a negative charge. Okay, not only are these two groups here hydrophilic, but they are also capable of making ionic bonds with things that are in solution, like other ions, or with each other when, say, you get a positively charged one interacting with a negatively charged one. All right. Another thing to think about is how we represent proteins. Remember, we are linking together a bunch of amino acids to make a full protein, right? So these little red arrows are indicating where one amino acid ends and where the other begins. Okay. So this part that is in the box, we call this the backbone of the protein because it has the same repeating structure, right? For each one of these amino acids, we have the amino group, central carbon with the R group attached, and the carboxyl group. And then we just repeat that. Amino group, central carbon, carboxyl, and with a different R group. Okay, so that stays the same. So a lot of times when we are writing out the overall shape of a protein, we don't necessarily write those atoms out just to make it less confusing. So down here we have three different ways to look at a protein. Excuse me. This one here is showing all the different atoms, and as you can see, it's a real mess. This one's just showing the backbone and not the R groups. Okay. And that's what this purple line and the yellow line and the in-between ones. And then this one is a space-filling model that really makes it look globular, which is a lot of what proteins end up looking like to our minds. All right, so we, in order to understand these complex proteins, so these molecules are really big. They can have hundreds of amino acids linked together to form the protein, or thousands of amino acids linked together to form the protein. And so they make these really large molecules that are folded up in interesting ways that kind of look like globs. And so in order to help us understand how proteins are put together, we divide up the structure into these 4 different levels of structure. Okay. Of course, we're just talking about one protein molecule when we're talking about all these structures. It's just a way to understand it. Okay, so there's primary structure. We abbreviate that with a 1 with a little degree symbol. So if you see that, it means primary structure. Same with secondary structure, tertiary structure, and quaternary structure. Okay, we're going to go through each one of these and talk about what it is and how it relates to the protein. So it's important to note that each level of protein structure is going to build upon the previous level to make these complex, large molecules that, remember, do lots and lots of work inside cells. All right, so what is primary structure? Primary structure is basically what we have talked about proteins up till this point. Okay, it's the sequence or order of the amino acids, okay, and they are held together by those peptide bonds, or remember, the covalent bonds. So I say a peptide bond, that's just a special name for the covalent bond between amino acids. Right, so we create the primary structure is created through dehydration synthesis, forming those covalent bonds. Okay, here it is written out, kind of like making you think of beads on a chain, which is a good way to think about the primary structure. And each amino acid is listed with its 3-letter code. Okay, we can also just list this as a list. So the primary structure, another way to write that would be to write met, phe, thr, asp, and those of course all stand for different amino acids: methionine, phenylalanine, threonine. You don't need to memorize those, but you do need to recognize what that's representing. Those are representing amino acids linked together, okay, creating the primary structure. So every protein has a unique primary structure. Okay, there are thousands of different types of proteins at work in cells all the time, and each has its own specific primary structure or sequence of amino acids. And next up is the secondary structure. So that primary structure then feeds into creating a particular secondary structure. Okay, these are highly regular local substructures in the backbone. So in any given protein, you might have some places of a certain type of secondary structure, and in another area have another type of secondary structure. There are 2 types of secondary structure that are recognized, and we call these helices or pleated sheets. Of course, when I went to college, we called these alpha helices and beta pleated sheets, so sometimes that's what comes out of my mind. Okay, so there's the primary structure, and that's feeding into the secondary structure. So this is just showing the backbone or the overall shape of the protein, okay, as these amino acids are folded up together. Okay, so there's the alpha helices, and here's the beta sheet. They also these areas that are sort of in between or that don't have any secondary structure, we call these random loops. Okay, different proteins will have different amounts or types of secondary structure. Like you could, for example, have a protein that has a bunch of alpha helices and no beta sheets at all. Or the reverse, you could have a protein that's all beta sheets. Okay, you could also have a protein that's mostly random loops. Okay, so how is this secondary structure created? It's created when parts of the backbone of the protein's backbone form hydrogen bonds with each other. Okay, let me switch color on my pen to make it show up a little bit better on this slide. Okay, so here we have the backbone, right? And this is actually coming from this direction. So nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. That is the backbone of the protein. Okay, and so this is written backwards to how we normally would, but there's just one amino acid, right? And here's a second amino acid, and so on. And here's the other side with its also backbone. Right? So this is part of a very long molecule that folds back on itself. Okay? And then it forms these hydrogen bonds. Remember, hydrogen bonds are shown by a dotted line. Okay? So all of these are hydrogen bonds. And what they're forming is between hydrogens on an amino group or the carboxyl group with its double bonded oxygen. Those are forming the hydrogen bonds. Okay. The important thing to remember is that we're talking about hydrogen bonds forming between parts of the backbone. Okay. Alpha helices do the same thing with even the same parts, but the result is different. Whether or not a protein forms a alpha helix or a beta pleated sheet has to do with, one, the environment that it's in and how it folds, and also what R groups are there. So, for example, if these two R groups that aren't shown in their full amount were actually repelling each other, say they were both negatively charged, then you wouldn't be able to get this formation there. Okay, so it's highly complex. And next up is the tertiary structure. So the tertiary structure describes the three-dimensional shape of the protein, or how that protein with its primary structure and secondary structure then folds up on itself to make this kind of glob. And you know, they have particular structures like little places that go in, or sometimes they have holes through them. Okay, so this is the tertiary structure. Okay. And each protein has a unique shape. Here are just some examples of different proteins that we find inside cells. I don't know what all of these do. There's a ribosome. It makes proteins, actually. Some of them form capsules where they can hold things inside of them. Other things have holes in them where they can allow proteins to go through, or things like ions or hydrophilic molecules to go through membranes. But the thing to look at here is every one of these has a different shape. Okay, and that shape is, of course, determined by the primary structure, right? Because what amino acids are there is going to impact how this folds, and I'll explain how that happens in just a second. And also importantly is that shape determines function. So for all of biology, function follows after form. So things have a certain shape in order to perform a certain function. Okay, and that's very true of proteins. So these proteins aren't just randomly shaped, they're shaped like they are so that they can do their function. Okay, so let's talk about how tertiary structure is formed. Tertiary structure is all about R group interactions. Okay, so we're not talking about parts of the backbone anymore. And what we are talking about are the R groups that are coming off. Okay, so there are 4 different types of R group interactions that hold the tertiary structure together and cause the tertiary structure. Okay, so you can have ionic bonds, right? And that's between 2 of those R groups that I showed you before that have a charge on them, opposite charges. They'll come together and form an ionic bond that will bring this part of the protein together, right? And cause it to fold. Okay, so we also talk about protein folding when we talk about protein shape. Okay, there's also these things called hydrophobic interactions. Hydrophobic interactions are just like when you put a drop of oil in water, the oil molecules clump together because they're in water. And the thing to always remember is that when we're talking about biochemistry is there's water all surrounding this protein. Okay, so if some of the R groups are hydrophobic and want to get away from water, they're going to head into the inside of the molecule and hang out with each other together, and we just call those hydrophobic interactions. That's not a super strong bond, right? Like you could shake this up and move it, but they will go back to hanging out together as long as they're surrounded by water. Okay, and then there's also hydrogen bonds, and this is between different R groups, okay, not between the backbone, so it's different than what we talked about with the secondary structure. So hydrogen bonds can form between different parts of hydrophilic polar molecules, like is shown here. And then finally we have these disulfide linkages. The disulfide linkage actually forms a covalent bond between 2 of the R groups that have sulfur in them. Okay, so in order to have that, you would have to have some of those sulfur-containing R groups. Okay, so you can see how this was, if you just look at the primary structure, it would be the sequence of amino acids, and that's not shown here, but each one of them would have its own particular R group. So here's the R group from one here, and here's the R group from the other. Okay. And the fact that they are attracted to each other is going to pull the parts of the strands of the protein together, and same here. Okay, and you get this overall folding. Okay, so importantly, R group interactions are dictated by the primary structure. Primary structure is, of course, the sequence of amino acids, right? So every amino acid is going to have a different R group. So if you have a different primary structure, you will have different R group interactions. Okay. The R group interactions cause the protein to backbone to fold in specific ways. Okay, and this leads to the specific shape. And as I said before, the shape determines the function. Okay. All right, so we've talked about primary, secondary, and tertiary structure. So primary structure is the sequence of amino acids. Secondary structure are local areas of twisting into helices or folding into beta-pleated sheets. And then tertiary structure is how that thing comes together and makes a specific shape. And the last level is quaternary structure. Okay? Not all proteins have quaternary structure. All proteins have primary, secondary, and tertiary structure. Okay, but only some proteins have this fourth level of quaternary structure. And these are proteins that have subunits. And the quaternary structure describes how those come together. Okay, so if you could imagine here that this little this is a subunit of a bigger protein. So some proteins will need multiple subunits, each with their own primary, secondary, and tertiary structure in order to come together and be functional. Okay, and those are the proteins that have this quaternary structure. All right, I'd like you to watch this video. I can't play it within the Panopto recording. It just doesn't work. So I'm going to have you watch it on your own, but it reviews everything we've talked about. It's really important. It's really informative, so please don't miss watching it. You can also just find a straight up link if you go to useful stuff on our Canvas website and go down to proteins, and then it's labeled, I think it's called, what are proteins? Something like that. Okay, so make sure you do that. Okay, so to review, all proteins have a primary structure, skipping over the secondary structure, which is important, but focusing on the fact that the primary structure gives rise to the tertiary structure. And remember, the tertiary structure is the shape of the protein or the form of the protein. You can think about it either way. And that then leads to the function or the job that that protein can do. Okay. Remember from our protein lecture, there are many jobs that proteins do inside cells, right? They're enzymes, they're transporters, they're part of our immune system, they're signals. They just do everything that cells need to do, and in order to do that, they need to have a specific shape. Okay, so it stands to reason then, if primary structure gives rise to tertiary structure, and that gives rise to the function, that if you change the primary structure, meaning you change the sequence of amino acids, you will then change the tertiary structure of the protein and change the function. Okay, and this would happen through a mutation in the DNA, and we're going to talk about that later when we talk about DNA. So right now we're just going to focus on this, like not how we get a change in primary structure, but if we do get a change in primary structure, what is the downstream effect for that protein? Why does its function change? Okay, and we're going to look at that within the context of a disease called sickle cell disease, or SCD. Okay. Sickle cell disease is caused by a mutation that changes the primary structure of hemoglobin. Okay, so first of all, hemoglobin is the molecule that's in our red blood cells that carries oxygen to our cells. Okay, it's vitally important for our bodies to be able to function. As we're going to learn later in the quarter, we all need a massive supply, constant supply of oxygen to our cells to fund cellular respiration, to make ATP, and if we don't get that, then our cells run out of energy and die. Okay, so hemoglobin is important. Hemoglobin has primary structure, so it has a sequence of amino acids for each of these. It has areas of secondary structure, so you can see here's alpha helices, so it has secondary structure. It has tertiary structure, of course, because everything folds in on itself. And then this is also one of the proteins that has quaternary structure, right? So there's 1, 2, 3, 4 subunits that come together. Okay, and really there are two subunits that are called alpha chains, and they're identical to each other, meaning they have the same primary, secondary, and tertiary structure. And there are two beta chains that have primary, secondary, and tertiary structure. Okay, these fold in a specific way to hug little atoms of iron. Okay, iron has a very strong affinity for oxygen. Okay, so iron is what allows hemoglobin to pick up oxygen and carry it to different parts of your body to deliver it to your cells. Okay, if you're anemic, that means you don't have enough oxygen, or excuse me, if you're anemic, that means you don't have enough iron, and your hemoglobin molecules can't form their necessary structure because they don't have the iron, and then you can't, carry enough oxygen to your blood. Carry enough oxygen to your muscles and to your brain and that kind of thing. Okay, so hemoglobin, in order to do its job, it needs to have this particular structure. Okay, so sickle cell disease is caused by a single amino acid substitution in the protein of hemoglobin. Okay, so sickle cell disease is an inherited disease. So a person who has it got it probably from one of their parents. Okay. So it's a change in the DNA, and we'll come back and talk about that when we talk about DNA. Okay, so this slide has a ton of information on it, so let's parse it out a little bit. I guess I'll keep with red. Okay, so first of all, let's just look at normal hemoglobin. Okay, so normal hemoglobin is functional, carries oxygen to your cells, and red blood cells are just packed full of hemoglobin. Okay. The primary structure, or the sequence of amino acids for normal hemoglobin, starts like this. Now, and this is actually only in one of the chains. It's the beta chain. There's about 140 amino acids here, but it only lists the first ones because that's where the that substitution or the change happens. Okay, so in normal functional, functional hemoglobin, the primary structure should be valine, histine, leucine, threonine, proline, glutamic acid, and glutamic acid again. Okay, and that forms this nice beta subunit that can then join up with the other subunits and make these lovely hemoglobin molecules that carry oxygen around. Okay, so this is the R group for glutamic acid, right? So that's this guy's R group right there. Notice that you would classify this as hydrophilic and ionic. Okay, so it's going to like to interact with water, and if it is in a protein, it's going to be poking out towards the outside of the protein, interacting with water. All right, so now let's look to see what happens to the primary structure with sickle cell hemoglobin. So sickle cell hemoglobin has the beginning that's the same, but at the number 6 amino acid, instead of glutamic acid, it has valine. Okay, valine looks like this. This is the R group for valine. Okay, how would you classify that? It's only got carbon, carbon, and carbon hydrogen bonds, so it's very hydrophobic. And I'll write hydrophilic over here. Okay, hydrophilic things like to interact with water, make hydrogen bonds. Hydrophobic things don't. Remember, they're the ones that act like oil in water. So when they're in part of a protein, instead of that R group facing out towards the water, it's going to be pulled in to the inside of the protein. Okay, and this causes a change in the tertiary structure of that subunit. Okay, so instead of looking like this, it now looks like this. Okay, it still joins up with its other subunits, but it then makes this quaternary structure of this protein that has these little hydrophobic parts here that are in water, and they get with a bunch of other hemoglobins, and they form these long fibers. Okay, these long fibers are really bad. Okay, so the molecules themselves can't. They have a diminished capacity to carry oxygen, and then they form these long fibers. Okay, and that has the result of causing the blood cells to take on a sickle shape, which is where you get the name sickle cell anemia. Okay, so looking up here, first at the normal red blood cells, they have only normal hemoglobin. Okay, and all of them form these nice round discs. Okay, these are very bendy, so they can fold, and they're very flexible. That's a really important function of the red blood cells, because when they get to your tissues, like way into your little tiny parts of your finger or other places, the capillaries are really, really small. They get down to where they're only 1 cell thick, and they have these branches in them. Okay, normal red blood cells can navigate those narrow, small passages. They can squeeze through them because they're flexible and deliver oxygen clear to all of your tissues. Okay, sickled red blood cells have lots of sickled hemoglobin. Okay, and remember, the sickled hemoglobin forms those long fibers. Okay, so in here we have long fibers of hemoglobin, right? So remember that from right here, the long fibers. Okay, and that pushes the edges out. So instead of having a nice round shape, we now have what's called a sickle shape, which gives it its name, sickle cell anemia. Okay, and these red blood cells, aside from not carrying oxygen very well because that hemoglobin doesn't work very well, they also, their shape is very problematic. When they get down into those little narrow passageways, they cause clots. Okay, they get stuck at branches, and that causes the blood to back up. It makes it so there's no blood getting down to the extremities or to little parts of the brain or the heart. And so when somebody has sickle cell anemia, it's like they don't have enough oxygen. Parts of their body are being starved of oxygen, and it's very painful. So there's a lot of extremity pain. There's ultimately organ failure. It can be hard to think straight because your oxygen's not getting to your blood enough, or getting to those parts enough. Okay, so all of this disease is caused simply by one single change, from a single amino acid substitution change in the primary structure. So we see that a change in primary structure leads to a change in tertiary structure, or the overall shape, and then that leads to a change in the function of that molecule. And that's, the real big theme with proteins. Shape matters. The primary structure, sequence of amino acid, dictates the tertiary structure, and then if you change the primary structure, you're ultimately going to change the tertiary structure and change the function of that molecule.